Hello everyone, and welcome to this video on agricultural plant physiology, where we will discuss the C3, C4, and CAM pathways of photosynthesis. Let's dive into the topic and understand these different mechanisms of carbon fixation in plants. Introduction to dark reaction. Photosynthesis consists of two phases, the light reaction and the dark reaction. The dark reaction is the process where CO is converted into sugars. It occurs in the stroma of the chloroplast and is also called carbon fixation or carbon assimilation. Despite its name, the dark reaction can occur in the presence of light as well. There are two main pathways of carbon fixation. 1. Calvin cycle, C3 cycle. 2. Hatch and Slack pathway, C4 cycle. Let's explore them one by one. C3 plants, Calvin cycle. The Calvin cycle was discovered by Melvin Calvin in the unicellular green alga chlorella, for which he received the Nobel Prize in 1961. Since the first stable compound in this cycle is a 3-carbon compound, 3-phosphoglyceric acid a 3-PGA, it is known as the C3 cycle. Characteristics of C3 plants. 1. Found in all photosynthetic plants, including hydrophytic, mesophytic, and xerophytic plants. 2. Photoactive stomata. Stomata remain open during the day. 3. High rate of photorespiration, leading to lower efficiency in CO fixation. 4. Normal leaf anatomy, no specialized structures like Kranz anatomy. 5. Energy requirement, to synthesize one molecule of glucose, 6 CO fixation, 12 NADPH and 18 ATPs are required. 6. Single CO fixation, Colorado is directly fixed by RUP, ribulose 1,5-bisphosphate. 7. First stable product, 3-PGA, 3-phosphoglyceric acid. 8. First enzyme involved, Rubisco, ribulose 1,5-bisphosphate carboxylase, oxygenase. 9. Carbon dioxide compensation point, 3070 ppm. Examples of C3 plants. Cereals, rice, wheat, barley. Cash crops, cotton, tobacco. Legumes, soybean, pea. Root crops, potato. C4 plants, Hatch and Slack pathway. C4 plants use an alternate pathway for CO fixation, where the first stable compound is a 4-carbon compound, oxaloacetic acid, OAA. This pathway is known as the Hatch and Slack cycle. These plants exhibit a specialized leaf anatomy called Kranz anatomy, where the vascular bundles are surrounded by large parenchymatous bundle sheath cells. Characteristics of C4 plants. 1. Found in tropical plants that are adapted to high temperatures and intense sunlight. 2. Mostly mesophytic plants, moderate water requirements. 3. Photoactive stomata. Stomata remain open during the day. 4. Negligible photorespiration, making the process more efficient. 5. Kranz anatomy, special leaf structure for effective CO fixation. 6. Energy requirement, 12 NADPH and 30 ATPs for one glucose molecule synthesis. 7. First stable product, OAA, oxaloacetic acid. 8. First enzyme involved, PEP carboxylase, phosphoenolpyruvate carboxylase. Examples of C4 plants. Cereals, maize, sorghum. Sugar crops, sugarcane. Millets, pearl millet. Weeds, cyperus. CAM plants, crassulacean acid metabolism. CAM plants belong to the Crassulaceae family and grow in semi-arid conditions. Their first stable product is malic acid, and they follow a unique mechanism where CO fixation occurs at night, reducing water loss. Characteristics of CAM plants. 1. Found in xerophytic, drought-adapted plants that survive in arid conditions. 2. Scotoactive stomata. Stomata open at night to minimize water loss. 3. Negligible photorespiration, making them highly efficient in water conservation. 4. Xeromorphic adaptations, thick, fleshy leaves to store water. 5. Energy requirement, 12 NADPH and 39 ATPs for one glucose molecule synthesis. 6. Double CO fixation, first at night and then during the day. 7. First stable product, OAA, oxaloacetic acid. 8. First enzyme involved, PEP carboxylase. Examples of CAM plants. Succulents, Opuntia, prickly pear cactus, bryophyllum. Tropical fruits, pineapple. Orchids. Conclusion. C3 plants dominate in cool, moist environments, but they suffer from photorespiration losses. C4 plants have crans anatomy and thrive in hot, tropical climates, making them more efficient in CO fixation. CAM plants are adapted to arid regions and use nighttime CO fixation to reduce water loss. These adaptations help plants maximize their photosynthetic efficiency under different environmental conditions.